Okay, so today we're going to start talking about the urinary system. And I don't know, like whenever we get into this urinary system, it's almost like feels like we're done. So we're very close to the end. We basically just have this chapter 24, and then we have a chapter 25, but they're going to be kind of, you'll see that a lot of stuff is going to overlap. And then the last unit is going to be on reproductive system. So it's two chapters, but it really feels like just one big topic. So just two more tests and a few more quizzes, and we are done. So let's talk about the urinary system. The urinary system is composed of kidneys, ureters, urinary bladder, and urethra. And it is a system mainly responsible for filtering of blood. In the process, it's going to remove metabolic waste from our body, and it's going to form urine. As most of the systems in our body, this is not the only function of the urinary system. So all our systems in our body usually have something else that they do. It is the truth for the urinary system as well. So urinary system has other functions, and these include blood pressure regulation, which we kind of already touched upon when we talked about blood pressure. And we talked about how blood pressure depends on the amount of fluid in your blood. The urinary system is also going to regulate ions and acid and base balance, which is basically just two special types of ions. It's going to be responsible for elimination of biologically active molecules like hormones and drugs. The urinary system also produces calcitriol, and this is going to increase your blood calcium levels. And it's going to also produce erythropoietin, which we've already mentioned in chapter 18 when we talked about blood. Erythropoietin is a hormone that is going to, it's a really a stimulating factor that's going to stimulate formation of erythrocytes. And so erythropoietin is necessary for erythrocyte formation. Kidneys can also perform gluconeogenesis, but gluconeogenesis is not regular function of the kidneys. And this generally doesn't happen unless like your body is going through severe starvation. But it can happen in the kidneys. So although not regular typical function, kidneys are able to do gluconeogenesis. All right, so let's switch to the next page here. And here we go. Okay. So what do you think here? Let's start this. And let me see if I can hide names. There. Okay, and I think I only have like a couple of people here. So answer it in your head if you don't want to do it interactively here. Structures of urinary systems are intraperitoneal. Do you think they're intraperitoneal or retroperitoneal? Okay, we have one vote for, I guess B is false, right? So one vote for no, and one vote for no is correct. So if you remember, when we first talked about what are intraperitoneal and retroperitoneal organs, I actually used kidneys as a classic example of retroperitoneal organs. So they are sitting behind the peritoneum, and this is going to be true for the whole urinary system. The whole urinary system is outside of the peritoneal cavity. So it's lined by the peritoneum only from the anterolateral portions. All right, so let's look at this. And this is the inferior cross-sectional view here. So you're looking from the bottom, right? And anterior would be at the top of this image, okay? So it's kind of an odd section here, but you can see on the whole body over there in the upper left corner where the section is done. So that's what this would look like here. And that's about it. But if you look at the peritoneum here, which is it even shown here? It's not really shown, but you could see the peritoneum, how it's lining, like especially in the anterior, how it's lining the cavity. And then when it gets to the kidney, I wish I could, hold on, I can annotate this. Let me see. When you get right here, like this is your, oh, why did it pick this color? Okay, it's kind of yellow. I hope you can see it. See, the peritoneum goes like this, okay? And so this part right here is well, that's liver, and liver is intraperitoneal. Okay, so all of this is intraperitoneal, and then all of this, the kidneys, are right behind that lining. Okay, so hopefully you can kind of see how that works there. And let's get rid of the annotation tool, and let's go on. Come on. There we go. Okay, we're going to talk about the internal anatomy of the kidney. This is really important, so make sure that you are able to label the different parts of the kidney. Internally, kidney is going to be divided into 3 distinct regions, although usually we say like 2 of the physical regions. I like to talk about the space as kind of a 3rd region here. So you have an outer cortex, you have a medulla, and then you have the renal sinus, and renal sinus is that space. Cortex is going to be the area where the renal corpuscles of nephrons will be found, but we'll talk about those a little later. Cortex projects into the medulla and forms renal columns, and these renal columns are going to separate these conical renal pyramids of the medulla. So the renal pyramids, they look on the flat picture, look kind of like a triangle, but they're actually a structure, their structure is like a cone. So the pointy part is going to point towards the sinus. That's going to be your renal papilla there. There are about 8 to 15 renal lobes, and each renal lobe is going to contain a renal pyramid. It's going to have parts of the adjacent renal columns, adjacent meaning next to them, next to the renal pyramid, and then also contains the cortex above the pyramid. So that section that you can see highlighted here, do they even label that as a lobe? Okay, let me make sure that you see what I'm talking about. But the renal lobe would be this. Okay, so all the way here. Okay, all this. So it has the renal pyramid, parts of those renal columns on the sides of the renal pyramid, and then also the cortex belongs there as well. So that's your renal lobe, and we have 8 to 15 renal lobes like this. The renal sinus is not part of the parenchyma. Parenchyma is that is a functioning tissue or functional tissue. The renal sinus is going to be the space that collects urine. The urine will exit the renal pyramid at the renal papilla, which is that pointy part of the cone that's oriented towards the sinus. It will enter the space that's directly surrounding the papilla, and this is known as the minor calyx. Minor calyces of adjacent pyramids will fuse together, and they will form these major calyces, and then major calyces will drain urine into the renal pelvis. So renal pelvis is the large space at the hilum of the kidney. Hilum is that concavity, and this will send urine into the ureter. So renal sinus is divided into three spaces. First, it's going to be the minor calyx. Then minor calyces merge together into major calyx, and then major calyces will lead urine into the renal pelvis. And all those 3 together form that renal sinus. And then urine will go into the ureter. And then from ureter, it will go into urinary bladder. And from bladder, it will go into urethra and then out of the body. So I've mentioned before the nephron. And by the way, something else that you can see here, a note that you can see the renal artery and renal vein. These are the blood vessels that are responsible for blood flow through the kidney or blood supply, right? So renal artery brings blood to the kidney. Renal vein is going to take away blood from the kidney. Okay, let's, before we go into nephron, let's select all that are part of the lobe, one lobe of kidney, if you remember. So that lobe of kidney, what was part of one lobe of kidney? And you can pick several things here. But you shouldn't pick all of them. So think about what I kind of attempted to highlight with my touchpad on my computer. Okay, and I have a vote for, yeah. So we have renal pyramid, yes. So renal pyramid is part of one lobe of kidney. We have one vote for C, which is part of the renal column, and that is also correct. So those columns on the side of each pyramid are also part of the lobe. And then E is renal cortex above the pyramid, and that is also correct. All the other stuff is going to be either on the outside of the kidney or part of the sinus, like minor calyx. That's part of the sinus. So it's not part of the lobe. Sinus is not part of the lobe. All right, good job. Well done. All right. So I've mentioned the nephron, the word nephron. Nephron is going to be the functional unit of the kidney. It is composed of two main parts. You have the round renal corpuscle and then the long renal tubule. The round outside of the corpuscle is created by parietal layer of the glomerular capsule. And these are going to be squamous epithelial cells. There is a vascular end and the tubular pole at each renal corpuscle. So you have the vascular end is where the blood vessels enter, and the tubular pole is where the tube is exiting. So you can see that on here on this image. I'll have another nephron which has like a more detailed look at the nephron here. But again, the renal corpuscle is the round part, and then inside you see some blood vessels there. Well, there is a place where these blood vessels enter the renal corpuscle, and that's your vascular pole. And then where the tubule exits the renal corpuscle, that's going to be your tubular pole. Okay, so that's that, and yeah, so let's look at the detailed look at the nephron here. And this is also kind of an important image here for you to be familiar with because it has different types of cells, and a lot of the structure has to do with how the nephron functions, okay? So here you can still see the vascular pole and the tubular pole here. So you can, if you look at the top of the diagram here, you can see the bracket that says renal corpuscle. So the round part is the renal corpuscle, and then we have the renal tubule. This image shows you a stretched out nephron. Nephrons in your body are really not like this. The tubule of the nephron would be kind of like, imagine that you have yarn, like a long yarn, and then you kind of bunch it up. Okay, that's how the tubules of the nephron would be in the kidney. They are going kind of in all directions. But this is like stretched out so we can see nicely what is where here. Okay, so the large structure inside of the renal corpuscle is known as the glomerulus. Glomerulus is a network of loops of capillaries. So as you look at that, you can see that the capillaries kind of create these loops. It is supplied by the afferent arteriole with an A. A is going to go in, leading the blood into the glomerulus. And then it's drained by the efferent arteriole, E for exit. Okay, so the blood will go through afferent arteriole into the loops of the glomerulus, and then it will exit the glomerulus through the efferent arteriole. As the blood flows through these capillaries, fluid is pushed out into the capsular space. I want you to think back to when we talked about capillary exchange. As the blood was flowing through the capillary, there was fluid that was being pushed out into the interstitial space, right? This is kind of similar, okay? As the blood flows through those capillaries of the glomerulus, there's going to be fluid that's pushed out. This fluid is going to fill the capsular space. It's going to fill the space inside of the renal corpuscle. At this point, this fluid is known as filtrate. The glomerular capillaries are covered by cells that we call podocytes, and those are the orange cells that you can see on those capillaries. These cells aid in filtration, as we will see in a little while as we go over the animation, and they are the visceral layer of the glomerular capsule. So if you look at the glomerular capsule, notice that the glomerular capsule has two layers, right? You have the visceral layer, which are those orange cells, which are those podocytes. And then you have the parietal layer, and that's the squamous cells that create that round structure. Okay. Okay. So the tubule, let's look at the tubule. The tubule of the nephron is divided into three main parts. You have the proximal tubule, that's the one that's near the renal corpuscle. Then you have the nephron loop, which is going to be literally a loop that is going to descend into the renal pyramid. And then you're going to have the distal tubule. And distal tubule is after the nephron loop. So it's distal because it's far from the renal corpuscle. So if you don't remember your directional terms, this is AMP1, like unit 1, when you were talking about proximal and distal. So we're talking in reference to the renal corpuscle. The distal tubule will eventually attach to the collecting duct, and all parts of the nephron except for the nephron loop are in the cortex. So the whole nephron, except for that loop, is in the cortex. The nephron loop that descends into the renal pyramid and the parts of the nephron loop are going to be called, or they're going to be described based on direction of the fluid in there. So when you look at the nephron loop, the part of the nephron loop that goes down into the pyramid, that's going to be your descending limb, because the fluid is going to go down into the renal pyramid. And then there's that bottom loop. We call that a hairpin turn of the nephron loop. That's not really labeled on this picture. And then you have the part that's going to turn up, like it's going to turn upwards, and this is going to go back towards the cortex. That's ascending limb. And they have, both of these limbs, both of these sections have thick and thin segments, and the thick segments are going to be cuboidal cells, and the thin segment is going to be made out of squamous cells. As urine is formed, there will be a lot of movement of fluid and substances across these tubules. And that's what I'm talking about, all the tubules, like the proximal, the nephron loop, and the distal tubule. So the animation that we're going to look at will hopefully help you understand how things are moving back and forth, okay? Because it's not going to be as simple as we saw with the capillary exchange. So compared to capillary exchange was very simple compared to this. So there are two types of nephrons when we look at this next slide here. And they are, there are nephrons that have renal corpuscles close to the medulla, and they have very long nephron loops. And these nephrons are known as juxtamedullary nephrons. These are nephrons that are going to maintain really high concentration gradient in the medulla, which is very important to drive movement of fluid and ions across the tubule and between the tubule and the interstitial space there. But majority of the nephrons in our body are going to be known as the cortical nephrons, and these have short nephron loops, and their renal corpuscles are farther away from the medulla. So when you look at this image here, the one on the left side, that's the cortical nephron. Most of it is going to be in the cortex. And then the one on the other side is the juxtamedullary nephron, and that one has long, deep loop into the medulla. One thing that you can think of when you're trying to remember which one is which is that the juxtamedullary nephron, that's the long name. It has the long loop, okay? Long loops go with the long name in this case. Other thing that I wanted to kind of point out here that you can see well on this picture is the blood vessels that are surrounding that long nephron loop of the juxtamedullary nephron. Notice how they're kind of like almost like a ladder, kind of like a hanging ladder, right? And this blood vessel network, it's a network of capillaries. This is known as vasa recta. So when we talk about blood flow through the kidney, I will mention vasa recta, but vasa recta is basically capillaries that are surrounding the nephron loop of the juxtamedullary nephron. The cortical nephron, I'm not sure why they didn't show you any blood vessels or any capillaries around the nephron loop of the cortical nephron, because it would have some. They're just not arranged like that, and we don't call it vasa recta. We just call it peritubular capillaries, like the rest of the capillaries. But there would be some blood vessels here, so imagine them into the picture, okay? Okay, collecting tubules are short tubules that are connecting the distal tubule to the collecting duct. The tubular fluid will flow through the collecting duct into papillary duct, which is the part of the renal papilla. So this is going to be a very short tube. Once the collecting duct enters the renal papilla, which is that pointy part of the renal pyramid, it becomes papillary duct. We just changed the name of it because of where it's located. Here the fluid is now urine. So once the fluid enters the papillary duct, we call it urine now, and it will exit into the minor calyx, then from minor calyx into major calyx, and then renal pelvis, and then ureter, and so on until it eventually goes out of the body. No more modifications will be made to this fluid after it reaches renal papilla. Okay, so in the papillary duct, it's already known as urine, and that is whatever the consistency or concentration or composition of that fluid is when it enters renal papilla, that is what your urine will be like. There are two types of important cells that we have to talk about in the distal tubule and collecting ducts, or collecting tubules and ducts. And these are your principal cells, which bind aldosterone. Okay, so principal cells bind the aldosterone, and intercalated cells, these are going to regulate acid and base balance. Furthermore, intercalated type A cells, these are going to eliminate acid, and type B cells will eliminate base. So just remember that they eliminate, and then they eliminate the substance based on the type that they're on, like the first letter. So intercalated type A eliminate acid, A for acid, and then type B for base. Okay, so they eliminate base. Both eliminate the substance. Type A eliminate acid, type B eliminate base. So that's that. So let's look at something else that you might have noticed. Oh wait, never mind. Let's match some pairs. Okay, I'm gonna go ahead and look at my pairs here. So if you have this pulled up through Nearpod, then you see it as a matching activity. If not, you can see, actually, if I bring this up, then it tells you the answers. Never mind. Again, this will be part of the self-paced presentation. So okay, looking good here. 7 out of 7. 5 out of 7 here. And sometimes on my end, it's a little slow. Okay, good job. You matched the pairs. All right, let's look. And this image just shows you a little bit of histology here. So what you would be expected to see, I mean, what you should expect to see if you're looking at the kidney, you see a lot of cuboidal cells. And actually in AMP1, when we talk about tissues, a lot of times they use kidney for cuboidal cells because the tubules have a lot of cuboidal cells there. And then you can see here, okay, let me draw around this right here. I don't know how to change the color and I don't want to mess with it, but right here in the yellow, that is your renal corpuscle. And inside that, you see the kind of like a cluster of cells. It's what it looks like. That's your glomerulus. So that's the capillaries there. What you can see, like where I made my yellow drawing, let me remove it. But in that area, you kind of see space that's almost resembling a circle around the glomerulus. And that's that capsular space. So when you're looking at kidney, I would not give you like a picture of just tubules from the medulla. I would give you cortex, and you would see the renal corpuscles and with the glomeruli on the inside. I'm sorry, there are some dogs. And then on the other part, you see the renal medulla, but renal medulla is really just tubules. So, All right, let's keep going on. Let's look at juxtaglomerular apparatus. And juxtaglomerular apparatus is part of kind of a regulatory mechanism of the kidneys or of the well, it's of the kidneys. And it's located near the vascular pole of the renal corpuscle. So you perhaps noticed that there was a tubule at the vascular pole, okay, and you can see that as you see those two blood vessels there in the middle, between those two blood vessels, there is this tubule. This tubule is actually your distal tubule of that nephron. So when we saw it all stretched out, that it didn't really quite show you that the distal tubule actually comes when it comes up, it comes in contact, it goes between those blood vessels at the vascular pole. So it's the distal tubule of that same nephron. So it comes in contact with both efferent and afferent arteriole. This is part of your juxtaglomerular apparatus, and it monitors both concentration of sodium and chloride, and it monitors blood pressure as well. So it monitors two things, and they relate to each other. The important cells here are your glomerular cells and macula densa cells. The granular cells are not glomerular cells, granular cells. I'm sorry. So granular cells are smooth muscle cells of the afferent arteriole, and these are going to respond to stretch. They contract if stretch, and therefore pressure is too high. And they do the opposite if the pressure is too low. This might seem a little odd at first because I'm saying if the pressure is high, you're gonna see vasoconstriction. Like what? That doesn't make sense. But it actually does make sense for the kidneys because by contracting at that place, yes, in that spot where the contraction is happening, the pressure increases if you do vasoconstriction. But what that does is it limits the amount of blood that's allowed to go into the glomerulus, and it will reduce the blood pressure in the glomerulus. Okay. So and this will be part of the animation that we will go over, or you will go over. The granular cells are going to synthesize renin as a response to low blood pressure, and this is part of your renin-angiotensin system also. So if you remember, we talked about this in the blood pressure when we talked about blood pressure. So that's that. Macula densa cells are your modified cells of the distal tubule directly next to the granular cells. These cells respond to sodium chloride concentration, and they signal granular cells to release renin if necessary. If blood pressure is too high, the amount of sodium chloride in the tubular fluid is going to increase because there isn't enough time to reabsorb it. Higher blood pressure means the fluid is moving faster through the tubule. This will signal granular cells to contract and limit the amount of blood into the glomerulus. If the blood pressure is too low, macula densa will signal release of renin, and that will cause systemic vasoconstriction ultimately. So that's kind of how that works. You can see the macula densa cells are different. They don't look cuboidal. They look more like columnar cells. And they are right there where I keep pointing at it like you know, like you can see where I'm pointing, but that's they're coming in contact with those granular cells there. All right. So a few more things before we are ready for the animation. And since we had a delayed start, I'm thinking that I will just let you go over the animation on your own. There is a kind of a check your understanding thing that goes with the animation, and then you can break it apart into sections according to your schedule. All right, let's look at the blood flow here. Also very important thing. So I will ask you to, maybe put these in order and things like that. So make sure that you're familiar with the blood flow here. The blood flow, the blood is going to enter kidney, as I've mentioned before, through the renal artery. Renal artery will split into segmental arteries right as it enters the kidney through the hilum. Segmental arteries will branch into smaller interlobar arteries. These are going to run along the renal pyramids towards the cortex. So they're running along the sides here. Once reaching the curved part of the renal pyramid where the cortex begins, the artery branches into an arcuate artery, and it runs along the arch of the renal pyramid. That's why it's called arcuate for arch. And the arcuate artery has branches that are extending through the cortex. So they kind of shoot into cortex. And these are called interlobular arteries. Afferent arteries or afferent arterioles project from the interlobular arteries, bringing the blood into the glomerular capillaries. So now we are at the nephron part, right? So we basically described how the blood gets to the glomerulus, okay? So blood flows through the glomerular capillaries, and then it exits the glomerulus through the efferent arteriole. And efferent arteriole will very quickly branch into peritubular capillaries. It is here where gas and nutrient exchange happens between blood and cells of the kidney. However, many other things happen along these capillaries. A special name is given to peritubular capillaries surrounding the nephron loop of juxtamedullary nephron, and it is vasa recta, as I've mentioned in that previous picture, remember? So peritubular capillaries that are surrounding the nephron loop of juxtamedullary nephron are known as vasa recta. After blood flows through the peritubular capillaries and or vasa recta, it is then collected by interlobular veins, then arcuate veins. So now it's like we're going backwards, and there are veins. Then interlobar veins along the sides of the renal pyramid. And then these interlobar veins merge directly into a renal vein. There are no segmental veins. So make a note, no segmental veins in kidneys. Although from the picture, it looks like we could name some of these segmental veins, we don't. Okay, so the interlobar veins then merge directly into renal vein. So that's blood flow. But there's another thing that we have to kind of talk about when we talk about kidney, and that's flow of fluid, which I've already kind of talked about sections of it, but we didn't quite put it all together. So as the blood flows through the glomerulus, we said that fluid, it will filter out. And this fluid is plasma without the proteins. Okay, it's like your blood plasma, but not the proteins. So fluid is filtered into the capsular space. And this is called filtrate. Okay, we've already said that. The filtrate then enters the proximal convoluted tubule, and now it's called tubular fluid. It moves into the descending loop of the nephron loop, or a descending limb of the nephron loop, and into the renal pyramid, then goes through the ascending limb of the nephron loop into the distal tubule in the cortex. Then it flows through the collecting tubule, then collecting duct, then papillary duct, and then from papillary duct, it goes into minor calyx, and then we don't see that here because we stop here at the papillary duct. So you can see that in the papillary duct, we already call that fluid urine. Okay, here's that figure continued here. So after papillary duct, urine will flow into minor calyx, then it will go through major calyx, then renal pelvis, then from renal pelvis into the ureter. Ureter brings urine into the urinary bladder, and then urinary bladder, when it's filled with enough urine, or whenever you want to consciously expel urine, it will contract and expel urine into the urethra and out of the body. So that's our fluid flow. The urine formation, okay, if we look at this image right here, this shows you the processes that are involved in the formation of urine. There are three main processes that are important to think about. Filtration is the first one, and filtration is the process of pushing fluid out through the filtration membrane, which is in the renal corpuscle, into the capsular space. It's the actual fluid leaving the blood. During filtration, nutrients cross the filtration membrane, and you would, if I had you guess, you would probably guess that you don't want nutrients to be part of your urine at the end. So these nutrients will be taken back into our blood in the process of reabsorption. Not all molecules that we must eliminate are filtered out through the filtration process. And so we have to eliminate some things actively, and we call this process secretion. Okay, so things that we do not want to eliminate in the urine and we want to take them back into our blood, that's known as reabsorption. And substances that we actively have to get out of our blood that normally don't filter out, they're removed in the process of secretion. So this next page will take you to a website with an animation, and when you go over this animation, I actually rewrote all those questions from this animation into the Google form, and I would like you to first answer it in the animation because it will tell you if you're right or wrong. And then record your answers in the Google form, and this will count as an extra credit for you. Everybody should get 100 on this, 100%, because the animation, if you do it within the animation, you know what the right answers are, okay? So at least the 5 of you that are here, or I think I have Jose. Jose's not gonna do it, but so 4 of you that are in here should get all the points for this animation. So I'm gonna show you, I'm actually not gonna go to the next slide, but it would be right here. I'm gonna actually, I pulled it up in another tab here, and we're gonna look at overview, and you'll kind of see how this animation works. And then you have a link to this if you go to your MyTCC, go to your lecture units, unit 5, and it's at the very top, urinary animation. And then below that, you have urinary animation extra credit. That's that Google form. So make sure that you watch the animation and open both tabs at the same time. It's easier to do it that way. So also for the Google form, you will probably need to be signed into your browser with your MyTCC credentials. If you are not, it won't let you in, but just sign out of your personal Gmail and log in with your MyTCC, and it will go good. So go well. So let's look at this. I hope that the sound works for this. Okay.
The nephron is the functional unit of the kidney. Every part of the nephron carries out specific physiological functions that remove metabolic wastes and toxins from the blood and result in the production of urine. We will discuss what occurs in each individual section of the nephron. In addition, we will explore the factors and regulatory mechanisms that influence filtration and determine the final quality and quantity of urine. We will first focus on the process of glomerular filtration in the renal corpuscle. Next, we will look at the glomerular filtration rate, or GFR, and its regulation by appropriate homeostatic mechanisms. Then we will examine reabsorption and secretion in the proximal tubule, followed by countercurrent multiplication and exchange in the nephron loop. We will then investigate selective reabsorption and secretion in the distal tubule and cortical collecting duct, as well as the role of hormones. Finally, we will see how dilute urine and concentrated urine are produced in the medullary collecting ducts. Go to the next page to see the animation for step 1, glomerular filtration.
Okay, so this is the only part that doesn't have a quiz after it, and it was just an overview as what's ahead of you. So I really shouldn't have started with that one. I forgot we just have kind of an intro here. All right, let's see. Come on, load. Okay, here we go. So if it just takes a little time to load, okay, don't worry, it will eventually load. So this is, and I like this animation because it has very short segments. Like this next one, this really first actual topic is only 2 minutes long. And once you go through this animation, it really takes you through most of chapter 24. And so there's just a few terms that are not used in this animation that I would like you to know. But this covers majority of chapter 24. Okay. So let's look at this first one, and I want you to see the
kidney
quiz.
Contains over 1 million microscopic structures called nephrons. Nephrons are the functional units of the kidney, each one capable of filtering blood and producing urine. Urine production begins with filtration. This process takes place across the filtration membrane, a porous structure located inside the renal corpuscle of the nephron. Filtration produces a fluid called filtrate, which is similar to blood plasma without the proteins. The filtrate collects inside the capsular space of the renal corpuscle, and from there enters the renal tubule to be further modified until it eventually becomes urine. Filtration happens at the glomerulus, a ball of fenestrated capillaries located inside a hollow chamber called the glomerular capsule. A layer of specialized epithelial cells, known as podocytes, wrap their foot processes around the glomerular capillaries. Between the foot processes are narrow gaps called filtration slits.
Okay, so this is, I think this is a great picture of what those podocytes really look like. You see the cell body there, and then they have these projections. Those projections are known as pedicels. And then you have, they kind of, the neighboring cells fit into each other, kind of like fingers of your hands, where it's not like a exactly super, super tight fit, but it's pretty, those gaps are actually pretty narrow. This is an electron microscope of these cells. So those spaces between those pedicels are known as filtration slits.
If we examine a cross-section of a glomerular capillary, we can also see that between the podocytes and the endothelial capillary wall, there is a layer of extracellular collagen meshwork called the basal lamina.
Okay, this is a, so remember, if this is, if you're looking at my hands here, if these are those pedicels of filtration and they create those filtration slits, the look at this is if you were to cut this way and you're looking, you know, at this. That's what you're looking at there.
Together, the fenestrated capillary wall, the basal lamina, and the podocyte filtration slits form the filtration membrane. These components of the filtration membrane act as a series of finer and finer sieves, which allow only relatively small molecules to pass through. As a result, filtration is selective based on molecule size. Water and small solutes, such as electrolytes, nutrients such as glucose, or wastes such as urea, leave the blood and collect inside the capsular space to form filtrate.
All right, so that was that first section. Then there's the button that you can see here that says go to quiz. So you will click that. And our first question is a little bit of an interactive question here. It's asking which of the ions, molecules, or cells will filter across the filtration membrane. So what do you think? What will go across? What will leave the blood and go into the capsular space? You can put that in chat. Anybody? Anything? Okay, sodium. Okay, sodium looks small enough to pass through. Let's click it. Okay, good. Sodium was good. What else? Oh, this light. Okay, water and glucose. We got water. Water is pretty small as well. And it's going to create bulk of the fluid, right? And then glucose looks very small as well. Before I click glucose, because she will start talking here in a minute, you can see that erythrocytes and leukocytes are definitely too large to pass through any of the opening of the filtration membrane. Make sure that you're able to tell me what the three parts of filtration membrane are, and that's your endothelium of the glomerular capillary. You have that basement membrane, or basal lamina, I think is what she called it. That's the same thing. And then you have the podocytes, the filtration slits created by the podocytes. So anyway, so leukocytes and erythrocytes are giant. Okay, they simply look too large. Platelet also is pretty big. One thing that looks like it could pass through the filtration membrane based on size is the albumin and other proteins, right? Like the albumin looks like it could go through the fenestration, which is fenestration is right here, right there. This is an opening in that endothelium, right? So albumin looks like it could fit through there, but what's going to happen to it is it's going to get caught by either the basement membrane or the filtration slits. It simply won't be able to pass the other parts of the filtration membrane. So just like she said on the video, it's the three parts of the filtration membrane serve as series of finer and finer sieves. The dogs are saying hello. Okay, so clear the drawings and let's select glucose.
Well done. All substances you have selected are small enough to be filtered through the filtration membrane.
All right, so that's your first question. Then most of them are going to be multiple choice. Filtration slits are formed by cells called what? Podocytes. Good. So if you click podocytes, it will tell you if it's correct. Now, if you click something else, it will tell you it's wrong. So again, I want you to go over this and try to get the correct answer. Don't just click them randomly to get the correct answer. Try to really pay attention to it because that's when it will be meaningful to your learning. But if you get it wrong, then rethink, okay, why did I think that? And what is the correct answer? And correct yourself. And then record the correct answers in your Google form. Okay, so it's the podocytes. Filtration is selective based on and it is size, yes. Sometimes people want to say solubility in water, but that's not necessarily the determining factor because there are some large molecules that are soluble in water that would not be able to pass through. So it is really strictly based on size. And then the last question, filtrate collects inside of what? So filtrate collects inside. And yes, I have one vote for capsular space here, and that's correct. Sometimes I have students jump at the glomerulus and they're like, oh, it's glomerulus because they know that it's part of that renal corpuscle. So make sure that you're really paying attention to the wording here. We're looking for filtrate, and filtrate is the fluid that left the blood. It left the glomerulus and it fills the capsular space. And then you would go into the next animation. So I just kind of wanted to run you through this and see, show you how this will work. And you will just record, you can record the correct answers on a piece of paper if you want, or you can have both of the things open at the same time and record the correct answers as you're going through it. I don't know if I think I said when the extra credit would be due. I think it's just the day before exam, maybe. I didn't say here, but let's go with day before exam. Okay, so do it by kind of like when the Learn Smarts are due, but I wouldn't wait until the day before exam. I'm just saying that's the latest I want you to do it, but definitely if you have some time today, try to run through that animation today, first time, and you will probably need to revisit this. Because once you get into the middle of this animation where it talks about countercurrent exchange and multiplier, that stuff is what gets intense. And so you will definitely need to watch it a few times. Okay. It's not just like hear it once and you know it. I mean, nothing works like that. Right. So anyway, so that's all I have for today. And we still got done at the right time. So sorry for the delayed start there today, but we got it. And any questions about this? If you don't have any questions, then you're free to go, and I'll see you on Wednesday. You're welcome. Yes. So filtration takes place it's pushing the fluid from the glomerulus into that capsular space. Yes. So as the blood flows through the glomerulus, some of the fluid is being pushed into that capsular space. That's filtration. Yes. And then reabsorption and secretion, that would be in the tubules. All right. Good.
Thank you. Have a good
You're
day.
welcome.
Bye.
Bye. All right, Kayla, did you have any questions or did you just step away? I don't know.